Welcome back to Microbiology, Part 3 of Chapter 11, where we explore translation, the process of building proteins from the genetic information stored in RNA. Our learning goals for Section 11.4 are to describe the genetic code and why it's considered almost universal, explain the process of translation and the roles of the molecular machinery involved, and compare how translation differs in prokaryotes and eukaryotes. Translation is the process of converting an mRNA sequence into an amino acid sequence. In other words, turning the genetic code into a working protein. And we're going to see later that this follows the genetic code of 3 base codons. Translation happens on the ribosomes that are either free in the cytoplasm or attached to the rough endoplasmic reticulum in eukaryotic cells. Translation requires 4 key types of molecules: mRNA, tRNA, rRNA, and protein factors that guide the process. It's energy intensive, but absolutely essential for cell survival. The genetic code is made up of codons, each containing 3 nucleotide bases, and there are 64 possible codons in total. 61 of them code for specific amino acids, and 3 serve as stop codons. The start codon is AUG, so those are the 3 nucleotide bases, and this codes for the amino acid methionine, signaling where translation begins. Most amino acids are coded by more than 1 codon, and this is called degeneracy of the code. Also, the third position in many, but not all codons, is called the wobble position, and this allows flexibility in pairing, helping to reduce the impact of minor mutations. Importantly, the genetic code is nearly universal, so this means it's shared across almost all living organisms. So here we see the genetic code. Here is the codon table used to decode messenger RNA, and each triplet of nucleotides, 3 nucleotides, corresponds to 1 amino acid or stop signal, as we see here. To read this chart or this table, what we do is, let's say we're looking for CUG. We can check for the first letter here. I know it's C, so it's somewhere in this box. Then we look at the second axis over here. I said the second letter is U, so I know it's somewhere here. And then the third letter is on this axis. I said it's CUG, so the third letter is G. When I put those all together, I can see this is the codon CUG, and it encodes the amino acid leucine. So there are 64 total codons, and 61 of them specify amino acids. 3 of them are stop codons that signal when translation should stop. And for the start codon we mentioned earlier, that is AUG, and that encodes the methionine amino acid. You don't have to memorize this chart, although I would like you to memorize 4 of the codons. 1 is the start codon, so you should memorize AUG is where translation starts, and the amino acid encoded there is methionine. And these 3, the stop codons over here. So we have UAG, UAA, and UGA. Whenever you run into these or any of these 3 stop codons, translation will stop. And one of my former instructors taught me one way to memorize this is when you say stop to someone, you might be thinking, you are gross, you are annoying, you go away. So that's how they taught us, and that was really helpful when I was learning this. So we just saw that there are 3 bases, 3 nucleotide bases for each codon, and each triplet codes for 1 amino acid in the protein sequence. What amino acid is coded for by the codon AAU? So I know A is the first letter, A is the second letter, and U is the third letter, so there it is. And that is ASN, which stands for asparagine. What happens when a stop codon is reached? So we saw these were the 3 stop codons. So we'll see later that no transfer RNA corresponds to a stop codon. So we're going to see proteins called release factors that are going to bind and break down the complex, allowing translation to stop. And this is just for fun, but codons are very popular, right? You can buy like a stop codon t-shirt, you get a pop socket with a codon chart, or even a mug. Ribosomes are rRNA protein complexes that serve as the site of protein synthesis. In prokaryotes like bacteria, ribosomes are 70S, made up of a 30S and a 50S subunit. In eukaryotes, they are 80S, made up of 40S and 60S subunits. We're going to see that in each pro or eukaryotic cells, the small subunit is going to bind to messenger RNA, while the large subunit is going to bind to transfer RNAs and catalyze the peptide bond formation between amino acids. Multiple ribosomes can actually translate the same messenger RNA molecule simultaneously, and they form a polyribosome to boost protein output. A complete structure containing an mRNA with multiple associated ribosomes attached is known as a polyribosome or polysome. And this figure shows multiple ribosomes translating a single mRNA strand at the same time. This is a very efficient system, especially in bacteria where protein synthesis needs to happen really fast. So in prokaryotes, that's what's shown here, where transcription and translation are coupled. So here's transcription, right? I'm reading DNA and making my messenger RNA. And as soon as the mRNA is made, ribosomes can attach and start translation. So we're seeing that here. This leads to long chains of ribosomes on a single messenger RNA molecule. In eukaryotes, we can also form polyribosomes, but only after messenger RNA processing has happened and the mRNA has been exported from the nucleus. So in eukaryotes, we cannot couple transcription and translation because they occur in different areas. Remember that transcription happens in the nucleus in eukaryotes, whereas translation happens in the cytoplasm. But in eukaryotes, once the mature mRNA reaches the cytoplasm, there can still be multiple ribosomes attached, and they can still form a polyribosome. I also wanted to point out that if this is the 5' end of my messenger RNA in pink, this is the 3' end, the direction of translation starts here and ends over here. So it looks like this ribosome has been on the mRNA the longest. Technically, that should have the longest chain of amino acids, whereas the one that just latched on should have the shortest chain. But the lengths look kind of weird in our book, so I wanted to show you if this is the 5' and 3' end of messenger RNA here. In this other reference, the oldest ribosome that's been on there the longest should have the longest chain. The newest one should have the shortest chain of amino acids. During translation, each transfer RNA molecule will act as an adapter that links an mRNA codon to its corresponding amino acid. So at one end of the tRNA, we're going to see an anticodon that base pairs with the messenger RNA codon. At the other end of the tRNA is going to carry the amino acid that matches that codon. An enzyme called aminoacyl transfer RNA synthetase will ensure that each tRNA is charged with the correct amino acid. This is a key step for translation accuracy. And charging means at the end of the tRNA, they're putting down the proper amino acid that corresponds to the codon seen on messenger RNA. Here are different images of transfer RNA. Here you can see the folded shape of tRNA, which results from internal base pairing, so internal formations of hydrogen bonds. Here is the anticodon loop, and that is going to pair with the codons on the messenger RNA. And on the other side, we have our amino acid binding site at the CCA tail, and that structure really ensures precise decoding during protein synthesis. So anticodon, if I look more closely, is going to be complementary in sequence to the codon. The codon determines what amino acid is placed here. And we have other space-filling models or a very simplified model of tRNA. Let's take a moment to figure out what would be attached over here. Which amino acid would be attached? Remember, it's based on the codon. So the codon is GCC. I'm looking for GGC. So there it is. GGC is glycine. So glycine would be the amino acid attached there. What if the anticodon were ACC, where this is the 3' end and this is the 5' end? So if I pretend that this was A, C, C, then my codon would be complementary. So A pairs with U, C pairs with G, G. So UGG would be my codon. UG, I'm just narrowing it down. UGG, that would mean that tryptophan. So up here would be tryptophan if this were my anticodon and this were my codon. Let's do a quick overview of what we've learned so far. So we had our DNA, and in the central dogma, we are transcribing the DNA into messenger RNA. What is going to be transcribed is the template strand, also known as the antisense strand. And if I look closely, remember we're starting at the 3' end of DNA because we can only build messenger RNA from 5 to 3. So they read T, put down A, read A, put down U, and so forth. Here we have the 3' end of messenger RNA. Translation occurs by the ribosomes. The ribosomes will look for the end, the 5' end of the messenger RNA in eukaryotes, for instance, and build amino acids there. So every 3 nucleotides corresponds to an amino acid according to our genetic code or codon chart. And then eventually this will lead to the building of our proteins, which ultimately lead to the physical characteristics we see. All right, our next checkpoint, the first question asks, describe the structure and composition of the prokaryotic ribosome. So prokaryotic ribosomes are 70S, and they are made of 2 subunits, a 30S small subunit and a 50S large subunit. Each of the 2 subunits includes both ribosomal RNAs and proteins, and together they form the site where translation occurs. For the second one, in which direction is the mRNA template read? The mRNA is read in the 5 to 3' direction. And we're going to see soon that the corresponding polypeptide is going to be synthesized from something called the N-terminus all the way to the C-terminus. In the third question, describe the structure and function of a transfer RNA. So transfer RNA or tRNA is a small folded RNA molecule with complementary base pairing. One end has the anticodon that base pairs with the mRNA codon. The opposite end has a specific amino acid that's found at the CCA 3' end. And an enzyme called aminoacyl tRNA synthetase charges each tRNA with the correct amino acid. So tRNAs act as adapters, translating the nucleic acid code into an amino acid sequence. So translation in prokaryotes, like bacteria, begins with something known as the initiation complex. And this includes the small ribosomal subunit, our messenger RNA molecule, the initiator tRNA that is going to be carrying the first amino acid, methionine. In prokaryotes, in bacteria, methionine is slightly modified, so it has a formal group attached to the methionine. And a formal group looks like this. It's just a functional group attached to the amino acid. We're also going to have several initiation factors. These are helper proteins, and GTP for energy. GTP is similar to ATP. There's going to be a sequence in my messenger RNA called the Shine-Dalgarno sequence that helps align messenger RNA correctly with a ribosome. And once the start codon is recognized, the 50S subunit will join, forming the complete 70S ribosome, and we're ready to begin elongation. Finally, it's important that translation starts at the correct reading frame. And a reading frame is the way nucleotides in messenger RNA are grouped into sets of 3 bases, also known as codons, during translation. So I showed you this image earlier. Here's my messenger RNA, and I can see groupings of my codons, right? 3 bases makes up a codon. But if something happens and something is shifted, and this becomes my reading frame for some reason, everything would be thrown off. It's hard to tell when I don't have a codon chart in front of me, but I can use an example here with this phrase, the cat and dog, right? These are nicely 3-letter words, kind of like codons. If I were to shift my reading frame slightly, let's say I just split these 2 away for some reason, and I just shift everything over, Now my phrase doesn't make any sense. So this is the correct reading frame. This is the incorrect reading frame. It's similar in codons. If you shift your reading frame incorrectly, you would create an amino acid chain that doesn't make sense for the protein you're trying to build. All right, so let me show you what initiation looks like, what that initiation complex looks like when we have everything put together. We have our 30S ribosome, our initiator tRNA with methionine or formal methionine if we're in prokaryotes, bacteria. And then we have our initiation factors that are not shown, our GTP that's not shown, and our large ribosome, the 50S complex. And right after this is formed, we're ready to elongate. So that's what we're going to talk about next. Before we jump into elongation in prokaryotes, though, let's pause and look at eukaryotes. How does initiation differ in eukaryotes? There are a few key differences. So first is that initiator tRNA carries regular methionine, not formal methionine. So that was in bacteria. We'll see regular methionine in eukaryotes. Also, there's no Shine-Dalgarno sequence in eukaryotes. So instead, the ribosome is going to bind to that 5' cap. Remember that 5' G cap. Then it's going to scan along the messenger RNA until it finds that AUG start codon. Then when the large subunit joins, the ribosome forms that 80S complex and we're ready for elongation. Translation occurs in a repeating cycle at 3 sites on the ribosome, the A site, P site, and E site. And A stands for aminoacyl, P is peptidyl, and E is exit, and they're usually arranged in this order: E on the left, P in the center, and A on the right. So I kind of think of like Environmental Protection Agency when I'm trying to remember the order of how they're in the ribosome. The A site is where the new transfer RNA comes in, bringing that new or next amino acid that is encoded by the codon here. The P site holds the growing polypeptide chain, and the E site is where the transfer RNA exits because it no longer has an amino acid attached, right? That amino acid has been added to that chain, and so it needs to get recharged with a new amino acid. In the P site, we have an enzyme. Actually, it's part of the rRNA that's within the large subunit. The enzyme is called peptidyl transferase, and that forms the peptide bonds between the amino acids. This whole ribosome complex then shifts 3 bases down the messenger RNA, and that shifting is called translocation, and this requires energy from GTP. So earlier we had initiation. The next transfer RNA comes in through the A site, so they're showing you the midway point of elongation. This has been going on for a while. The next tRNA came in through that A site. This whole chain will be transferred onto that site through that peptidyl transferase enzyme, forming peptide bonds between this last amino acid and the new amino acid. When that happens, this will be uncharged or decharged, right? There's no longer an amino acid attached, so it's uncharged and it exits through that E site to get recharged. And this will continue down until we reach a stop codon, which we'll talk about next, what happens for termination. So when the ribosome reaches a stop codon, either UAG, UAA, or UGA, this will signal the end of translation because no transfer RNA can bind. Instead, release factors will attach to that A site, causing the completed polypeptide to be released. The ribosome subunits then detach and can be used for another round of translation. So earlier we had elongation, remember, and this was the E site, P site, and A site. During termination, this ribosome is moving forward by 3 nucleotides, basically by another codon that was here, but when this whole thing shifts forward, here is the stop codon in the A site, causing release factors to bind, and the polypeptide will be released from the P site. So it's released from that transfer RNA, and now the ribosome subunits will detach and be recycled. As I mentioned earlier, in prokaryotes like bacteria, transcription and translation happen at the same time, right, simultaneously in the cytoplasm, because they do not have a nucleus. So as soon as an mRNA is transcribed, ribosomes attach and begin translating it. This coupling allows bacteria to respond rapidly to environmental changes. In contrast, in eukaryotes, we separate these processes. Transcription occurs in the nucleus, and translation occurs in the cytoplasm. Here's a visual image of that. So in prokaryotes, we have a coupling of transcription and translation because there's no nucleus, right? Everything's in the cytoplasm. We're transcribing DNA to RNA, and as soon as that mRNA is made, we can have ribosomes attach and begin translating the mRNA to protein. But in eukaryotes, we have our DNA where it's being transcribed to a pre-messenger RNA. We have that 5' cap, the poly A tail, the splicing that will occur and make mature mRNA that's going to exit into the cytoplasm. Once we have our mature mRNA in the cytoplasm, the ribosomes can attach and begin translation. All right, so this is a nice table comparing translation in bacteria, those prokaryotes versus eukaryotes. So in bacteria, they have 70 S ribosomes. We have 80 S. Remember, for bacteria, they have formal methionine with that formal group attached, whereas in eukaryotes, just regular methionine. In bacteria, we have that Shine-Dalgarno sequence that's recognized by ribosomes, whereas in eukaryotes, we start from that 5' cap. And then in bacteria, they have coupled transcription and translation, right? Everything's happening in the cytoplasm. In eukaryotes, it's not simultaneous, right? Transcription happens in the nucleus, right? The nucleus is transcription. Translation is in the cytoplasm. Cytoplasm is translation. Once translation ends, most proteins still need some modifications before they're functional. And common modifications include either signal sequences that target proteins to specific locations, chaperone proteins that help with correct folding, proteolytic cleavage to cut off certain portions and activate the protein, or even chemical changes such as adding phosphate groups, methyl groups, or glycosylation, which means to add sugar molecules to proteins in order to alter activity or stability. And these steps fine-tune protein function and their localization. All right, the first checkpoint question asks, what are the components in the initiation complex for translation in prokaryotes? So we have that 30S ribosomal subunit that binds to the messenger RNA template. The initiator tRNA is holding formal methionine. We have initiation factors and GTP, which is our energy source. And then that 50S ribosomal subunit joins to form the complete 70S ribosome. For the second question, two differences between prokaryotes and eukaryotes. So in prokaryotes, we had that initiator tRNA, which carries formal methionine, versus in eukaryotes, the initiator tRNA carries regular methionine. For mRNA recognition, for prokaryotes, we bind to a Shine-Dalgarno sequence, versus in eukaryotes, they bind to that 5' cap and then scan for AUG. For the three active sites of the ribosomes, I put them over here at the bottom. So the A site or aminoacyl site, usually found on that far right side of the ribosome, accepts incoming charged transfer RNA carrying that next amino acid that corresponds to the codon. Our P or peptidyl site holds the tRNA and it's attached to that growing peptide chain. And then the E site or exit site releases that uncharged tRNA so it can be recharged with a new amino acid. Finally, what causes termination of translation? So that is our stop codons, right? Our stop codons are UAG, UAA, and UGA that enter that A site. Release factors will bind instead of tRNA. The polypeptide will be released from the P site tRNA, and then the ribosome subunits separate and they're recycled for the next translation event. All right, let's recap 11.4. Describe the genetic code. Why is it universal or almost universal? The genetic code is the system that translates mRNA codons, which are 3 base sequences, into specific amino acids. We said there are 64 possible codons. 61 encode amino acids, and the other 3 are stop codons, UAA, UAG, UGA, that signal termination of translation. Remember that the start codon is AUG, and that encodes methionine, marking the beginning of translation. And because nearly all living organisms use the same codon to amino acid mapping, the code is considered almost universal, and it's a reflection of the shared evolutionary origin of life. All right, for the second one, explain the process of translation, the functions of molecular machinery of translation. We have translation that converts the nucleotide code in mRNA into an amino acid sequence or a protein. Remember, the ribosome is made up of rRNA and proteins. That's going to bind to the mRNA and catalyze peptide bond formation. And tRNA molecules act as adapters that match each mRNA codon with its correct amino acid. Aminoacyl tRNA synthetases are the ones, or the enzymes, that charge tRNAs with the proper amino acids, and translation occurs in 3 stages. We had initiation, where those ribosomal subunits, mRNA and initiator tRNA, assemble at the start codon. Then we have elongation, where tRNAs bring in amino acids, peptide bonds form as the ribosome moves along the mRNA, and then termination happens when we reach a stop codon and release factors free the polypeptide and the ribosome dissociates. For the third one, I'm going to work with you on this third question on the next slide in a table format. So here's a table we can use to practice that last learning objective, comparing translation in eukaryotes and prokaryotes. See if you can fill in the blanks. Pause the video and the answer key will be in the next slide. All right, and this is the answer key. So check your answers with these here. The only one I haven't directly talked about is this last one, where prokaryotic translation is very rapid, especially since it's coupled with transcription, whereas in eukaryotes it's slower because it's separated spatially and temporally. Remember, transcription is in the nucleus, then translation occurs in the cytoplasm. And that takes us to the end of our third video. In the next video, we'll cover 11.5, mutations. We'll look at how changes in DNA sequence can lead to changes in phenotype.